Texas Lutheran University.
Okay, so today we're excited to have a TLU alum, Dr. Lauren Dobson, here with us. She got her Bachelor of Science in Biology on the molecular track in 2006. She also got a minor in chemistry and in history. She got a master's degree and has her PhD from Texas A&M, where she's currently at in the College of Veterinary Medicine. She'll tell us a little bit more about her educational background and her path through academia in her seminar. So let's welcome Lauren. So I'm really happy to be here. I was in this room for freshman biology. It looks exactly the same back in 2002. There's Dr. Levens. And it's great to come back. And a little history about me is I was here from 2002 to 2006. I did do the molecular biology track because I, at the time, was trying to do pre-vet program. So that really isn't offered here, so I had to do some courses like animal nutrition from Oklahoma State. Needless to say, vet school didn't necessarily work out straight out of TLU. I was a softball and a volleyball player, so double sport while doing some organic chemistry, biochem, not the best option, but if your GPA is a little low, you can still become a doctor. Just takes a little bit. So as most seniors, you really don't know what your next step is going to be. I wasn't sure, but I had applied for some grad school to get my master's, because I thought getting my master's and then I'll go to vet school. So I applied to A&M Kingsville, and I got in. They never really told me, but I got in the first day of classes, so I moved down there. And I was in the animal science department, and my emphasis was on equine quantitative genetics under Dr. Stephen Lukefar. He actually is more into naked rabbits. He created the naked rabbit, and he does a lot of work with Heifer International. And he created the naked rabbit to help feed third world countries because they breed quickly, they're a good protein source, and the reason they needed to be naked or hairless was because they can't handle the heat tolerance, isn't very good, so they created the naked one and they supplied them to third world countries to help breed up and supply some meat source for underdeveloped countries. So I wanted to do horses. Luckily, he happened to know the King Ranch quarter horse division manager at the time. So we went out there, met with the King Ranch head honchos, and they allowed me to do my master's project with them, looking at pedigrees with their breeding mares, their breeding stallions, and I did a historic genetic characterization of King Ranch quarter horses. I'll go into that a little bit. Quantitative genetics is basically looking at expected progeny differences, heritable traits, and then applied to like cattle herds and ranching and trying to find out the best way to get the benefits of heritable traits into your herd. And then I took a year off after my master's because my brain was kind of done. Wasn't sure what I was going to do. I actually worked on the King Ranch and got an internship, so I was still working on the ranch. I worked in the quarter horse unit and then kind of ventured to wherever they needed me to go. And so I really enjoyed my time there, but it was time to move away from Kingsville. So I moved back home because my grandpa was sick. But I gave a lecture at the King Ranch quarter horse barn, and they put my picture in their, they have the King Ranch of Institute of Ranch Management, and they have a biannual newsletter that comes out, and my picture is in there. Well, my advisor at A&M, Dr. Jim Durr, saw the picture, contacted the guy who was running the Institute of Ranch Management and asked, hey, so the King Ranch got a geneticist finally? He was like, well, yes and no. She was a master's student. He contacted me in like January of '09, was like, you want to get a PhD? And I was like, sure. I kind of missed school. It was weird. I do like learning. And he was like, well, do you like bison? And I was like, don't know a whole lot about them other than that they almost were extinct and now they're back. And so we'll talk a little about some horses, bison. It's not just about animals though. I'll get to some stem cells. There are a few surgery pictures in here, so I'll let you know whenever those come up, but they're not that bad. I know you just ate lunch, but you should be fine. So again, Animal Science Department, Quantitative Genetics, Pedigree Analysis. While I was at A&M Kingsville, I got to be a part of a ruminant sampling. So ruminants are cows. We looked at steers, and they had a cannula, which is basically a hole in the side of their stomachs. And so you get to put a stopper, kind of like a wine stopper in there. So we were putting different feed sources in a bag and putting it into their rumen. And then we would take it out and weigh the different samples and see how that was affecting their forage diet and if it was helping with digestive degradation. And then A&M Kingsville also has a Caesar Kleberg Research Institute, which is a very good wildlife program there. And I got to help out one of my fellow grad students. He collected wetland samples, and he wanted to identify the different invertebrates, like little tiny guys, within those samples. So I helped out with that. And then through the Institute of Ranch Management, I also got an advanced ranch management certificate. Basically, you just take different courses that they offer. It's not usually through the school. It's just extra courses that they do throughout the year. So you meet those guidelines, and since I was taking some of those classes already at Kingsville, I figured I might as well just finish off and get them. So it was actually really beneficial. So different schools offer a lot of different things whenever you're there, and so luckily with my master's, the ruminant sampling actually helped finish up whenever I got to A&M because the steers were actually at the A&M research facility. So just to talk about my master's research quickly, what we wanted to do was characterize by breed the influence of foundation, thoroughbred, and unknown ancestry. So the quarter horse actually stemmed from the thoroughbred back in the early 1900s. So they wanted to see how much thoroughbred was still left in there, and the King Ranch does actually have some appendix or thoroughbred quarter horses where the mom or dad was a thoroughbred. So they do have those, and the cowboys actually prefer them because they're a little bit taller and they can handle big steers for roping. So compute genetic relationship coefficients of the stallions and mares to Old Sorrel, Mr. Sam Pepe, and Pepe Sam Badger. So Old Sorrel is going to be this black and white picture, and he was the founder of the King Ranch line. So the main reason they wanted to know inbreeding was they did a lot of line breeding back to Old Sorrel because he was producing a lot of good foals. So this meant that they are taking own daughters of Old Sorrel and then breeding back to him. Yes, it is kind of inbreeding, but the fun thing is, the only difference between line breeding and inbreeding is whenever it doesn't work, line breeding is whenever it works. That's what they like to say. So the next generation of well-known horses for them are Mr. Sam Pepe and Pepe Sam Badger. Pepe Sam Badger is Mr. Sam Pepe's son. They produce a lot of good cutting horses. They have a lot of earnings, and a lot of people still want these same bloodlines in their ranch horses. So we wanted to see how much that of the studs and the mares have actually kept with their relationships to these. I had 74 mares to look at, 16 studs, and we went 8 generations back. So that's a lot of paperwork to look through, but it's a master's project, so you got to do it. So the main purpose was the ranch is going to use my data for planned matings for the future. They also don't want to go too far into using just numbers. They also go based on conformation and looks whenever they plan their matings. So this was just another tool that they could use. So I know this is a little busy, but we'll talk about it. So from the pedigrees, 8-generation pedigree is going to be kind of long and drawn out. So we made arrow diagrams that you could trace back all of the known horses that went back to Old Sorrel only. So all of these were at one point on the King Ranch. So I did that for every animal. And you can see Mr. Sam Pepe's here, Pepe, Sam Badger, and then Old Sorrel. You can see how many different offspring Old Sorrel had just by tracing back the arrows to him. So this one was Taquito Sugar. He was born in 1992, and at the time, he was the oldest stud on the King Ranch. I think they sold him. So his inbreeding, keep in mind, was only traced back to Old Sorrel, was at 1.9%. So that's good. Their genetic relationship to Old Sorrel was 14.8%, Mr. Sam Pepe 14.2%, and Pepe Sam Badger 25.8%. So in the end, we were showing that they were maintaining their genetic relationships while keeping inbreeding low. So they are doing a good job. They do bring outside studs in. So last year, the guy currently running it, James Clement, he asked if I could look at a potential stud that they wanted to bring onto the ranch to replace Taquito. Because at the time, at that point, he was getting up there. So some further research was, they call this stud the Boone. He's a pretty red roan, so they wanted to know same thing, inbreeding, genetic relationships. How is this stud going to help our mares and our horses? So here's the Boone, Pepe Sam Badger, Mr. Sam Pepe, and then Old Sorrel's here. He only has a few lines going back to him, but he has a very good pedigree for cutting, though. He's won some money. So his inbreeding was at 4.5%. Genetic relationships to Old Sorrel was lower at 3.4%, but he had higher relationships to Mr. Sam Pepe and Pepe Sam Badger at 17.8 and 27.6 respectively. So they still use my research, luckily, to help plan who they're going to bring in to help with as their other studs get older. So again, like I said, I took some time off, got contacted by my old advisor, Dr. Jim Durr. He does bison research, and I was in the Department of Veterinary Pathobiology. It was mainly the genetics genomics track. Now, the genetics program at A&M was interdisciplinary, and I'm pretty sure it is because it has to encompass genetics of plants, bacteria. It's rather large. So the way that it usually goes is you get recruited to come to A&M, you apply to the program, and then they'll let you come in and meet everybody and get recruited that way. Luckily, since Dr. Durr wanted me, I didn't have to go through that process. So I knew that I was going to be in his lab, because whenever you start, you have to find your advisor. Whoever has funding, you get to rotate in their lab, and that. So luckily, he knew that he wanted me, so I got to go. And through the veterinary school, they will pay your tuition at least. So they have to make sure that you have funding before you come. They're not going to stick tuition, fees, and everything else on you. Do you have to pay fees a little bit depending on if you get a fellowship or scholarship, then they will cover everything. But tuition is at least covered, so that is good. In grad school, fees are only like $1,000 at A&M. I say only, but I compare it to here, because this was my most expensive schooling. A&M Kingsville, about like $3,000 for the whole year-ish. So I was like, they told me how much for the semester, and I was like, that's it? And I was like, here. So when I was there, I had to earn my money, so I was a TA. So I TA'd Wildlife Diseases, Great Diseases of the World, a third-year veterinary class called Exotic Wildlife, primarily focused on deer, the deer industry, and we brought in a lot of veterinary wildlife vets in to talk to the vet students, and it was they learned how to dart the animals too. We did like a darting class, so it was pretty fun. And then Biomedical Genetics, and I got to lecture that class a lot, and I still do because I'm still at the vet school, and I've taught it a few times, and if my advisor needs me to, I go and teach it. And I really enjoy teaching that class. It's a good class. So Dr. Durr runs the DNA Technologies Core Lab. It was a sequencing lab. Right now it's not. But he does bison testing. So people will send in tail hairs from their herds. There are about roughly 500,000 bison in the United States, North America. if you didn't know. Texas does have at least 10 herds. There is one out near Lake Bryan that's about the size of 50. There's one at Caprock Canyon State Park, which I'll talk about in a little bit. It's the Texas State Bison herd, and that one started from Charles Goodnight, who helped bring the bison from the population bottleneck back in the 1880s. The urging of his wife, they went and captured some bison calves after their parents had been slaughtered, and they started raising them. So it's a cool little herd out there. It's really pretty out in West Texas. If you want to go, I think you should. So I was responsible for that lab, sitting invoices, sitting the results, making sure the testing got done. It's mainly done by microsatellites or a mitochondrial test. So we extracted the DNA from the tail hairs or FTA cards, which is just like dried blood samples. And then I would perform, the mitochondrial test was easy. It'll come back with either if you're bison or cattle, because they do interbreed. And then for microsatellites, we looked at purity microsatellites because we had a few that if you test positive for cattle microsatellite, then you would have integration. And then also like heterozygosity and diversity, and then we used them for parentage as well. And so I was responsible for that pretty much the second semester I started at A&M, so while I was taking classes, doing that was fun. So some other things with my PhD work was that I learned that if you have an interest in, is a good thing, is bioinformatics. So I did genomic sequencing. I had to learn how to script, write code. I really don't like computers, but I learned. And so, because I had files, sequencing files that were really big. A&M has a server through TIGS, or Texas A&M Institute of Genomic Sciences and Society. I also had help from Iowa State University on that because my PhD work was a big collaboration of Iowa State, USDA, University of Maryland. So my whole genome sequencing was done at Iowa State and at USDA Meat and Animal Research Center. I went to USDA MARC, which is in Nebraska, Clay Center, Nebraska. It's a big animal research facility, and I learned how to make my genomic libraries there. Dr. Durr wanted me to be very hands-on, and if you go into research, try to be as hands-on as you want to be. I would say learn as much as you can, because it'll make you just that much more of a viable candidate for jobs, and you'll just diversify your portfolio. I did some karyotyping as well. So basically, this would be my bison sample for my PhD. This is the X and Y chromosome. And then we also did our karyotyping for our bison sample. And I'll show that it had the same number of chromosomes as cattle. That's why they can interbreed. And this was just a fish staining of karyotyping with the fluorescence. And then we put them together down there. So that was something else that I learned. We also did some historic bison sampling at the Smithsonian. So I got to go and collect around 40 samples from the Smithsonian, and I'll show you two that I have in my presentation. And then how I extracted those, because I was working in a bison lab. We had bison DNA. How are we going to make sure it doesn't get contaminated with modern bison? So I'll talk about that. And then I also I'm on 2 papers. They came out this year that looked at drug residue studies in white-tailed deer. Since the white-tailed deer industry is now becoming more of like a ranching, they're breeding them for horns and everything. They're using antibiotics. There is no real known withdrawal period on antibiotics for deer. They're just basing it off of cattle. So this was with Texas Wildlife Department, Dr. Don Davis, some people, some other veterinarians. So we injected the drugs, and then we had to sacrifice, and then from the tissues we ran and saw how long the residues were out there. We did a 10-day, 21 and 30, I believe, or 10, 14, and 21. And most of the drug residues were gone after 10 days. So it was very big for the deer industry for that one. And then using my master's work, I was able to help out for a Stallion Reproduction, basically looking at testes deformation and if inbreeding was leading to that for someone who was on my committee. So through grad school, you can be a part of many different things as much as you want, as long as your advisor allows you to. So some of the drug residue was kind of after I graduated, but I also did one while I was doing my PhD. So my doctoral research, just quickly, I'm not going to go into too much detail. We provided an annotated de novo Bison genome reference sequence because there wasn't one. It is available on NCBI. The paper is in progress. We did a comparison of historic and modern Bison sequences. So basically, I mapped them, the historic sequences, to our reference sequence and looked at SNP or indel differences. And then a comparative genomic analysis among modern Bison, historic Bison samples, and then to domestic cattle. Because the main purpose of this, at least for my advisor, is to create a better integration test for Bison other than just mitochondrial. And at the time, we only have 14 microsatellites. 14 microsatellites through a 2.8, 2.6 genome is not a lot. So we wanted to make sure we're actually covering the genome for integration. And this pretty guy is my Bison reference animal. His name is Templeton, named after Dr. Joe Templeton, who did a lot of Bison research in the 80s and 90s. And I actually got to go to one of Ted Turner's ranches in Montana and collect Bison samples from him after we darted him. He is still alive. I think he's in Oklahoma now, though. And we wanted to make sure, the reason why we went to Ted Turner's is because he received Yellowstone National Park bison to do a USDA quarantine because of the brucellosis status. We wanted to make sure that because of the integration with domestic cattle, Yellowstone is one of the only ones that's known to be integration free. So we wanted a Yellowstone animal. So our best bet was to go to Ted Turner's ranch because he had had these animals for 5 years and they didn't have brucellosis. So hopefully they're making ways on that for brucellosis, and USDA wants to use the genome to help identify genomic resistance to brucellosis and other diseases too. So, brief history of Bison. They are based primarily on physical appearance. They have wood buffalo and plains bison. There's actually more subspecies than that with northern and southern. They had the bottleneck in the 1800s, almost complete elimination, and then the hybridization of domestic cattle with bison was done because they were trying to make a better meat source, because bison meat, to me, tastes a lot better than cattle. But whenever they bred them together, it didn't really happen. That whole better meat source didn't happen. So now we have a lot of advanced generation back crosses, and we can't really tell just by looking at a bison that is it integressed or not. Unlike these, where you can tell. So we can see bison mom, bison calf, and then dad is back here. This is actually an F2, and Charlet, I believe, was in her pedigree somewhere. So those we can tell, but modern bison, usually we can't just tell easily. So basically, our reference genome is 2.82 gigabytes. Again, total number of chromosomes and organelles, 31. Just comparing cattle, the human reference genomes, we wanted to make sure we were in the right range. Genes and pseudogenes, we have about 26,001 in bison, and then it has the breakdown down there. We're not going to go too into detail with that. And then our mitochondrial size for bison is 16,319. So we have a good reference. It's available now. People can start using it. We wanted to do our historic sampling. So in order to be a historic bison to be considered to get sequenced, you had to be before the population bottleneck, you had to be well documented, and the Smithsonian did that for us. So this guy is actually skull sample number 9. He was collected in August of 1856. He was actually collected by Hayden, who was one of the Smithsonian's kind of catalogers. And There's actually the Hayden Valley now in Yellowstone National Park, so it was named after him, and he was found in Yellowstone. So if you were to look at a bison skull now, you could probably tell that they're a lot bigger than this one. The fun story with this guy down here was this was actually our skeletal sample number 1. He was donated to the National Zoological Park by Charles Goodnight, and then it died in July of 1903. So we do have this sample that kind of can be a backdrop for the Caprock Canyon State Park to kind of gauge how it's changed over time. So I went to the UNT Health Science Center in Fort Worth. I actually worked with a forensics lab because we have very delicate samples. We need to know how it's the best way to extract these. So here's our sample. Put it into this lovely little tube with this metal bar grinder thing. Put it into a liquid nitrogen mill. It goes for about 10 minutes, and it'll turn this bone into this powder. And then from this bone, or this bone powder, you use like a spin column, QIAGEN column to extract the DNA from. And it's very pure, and since we did it in a human lab, we knew that it was bison, modern bison weren't gonna get in there. And then any of my PCR stuff that I did under a hood, so it wouldn't get contaminated that way. And also my DNA extractions. And then we would send the DNA off for the genomic libraries to get done at Iowa State. So I thought that was fun. The director of this lab is actually Dr. Bruce Bedoli, and he was the FBI director at one point. So he was a fun guy. So long story short, on all of my research and sequencing, had about 15 bison sequences that I got to look at because we did Caprock Canyons, Yellowstone National Park, and also 4 wood bison or wood buffalo. And this is how you would do the bioinformatics if you like computers. We basically get our sequences. We have to trim these reads based on quality and coverage. And then I mapped them using the server at TIGS, using a script to the UMD 3.1 thousand bulls reference genome because it has the most diverse cattle breeds within it, because I wanted to at least encompass all of the cattle breeds that bison could maybe breed with. So took all of them, aligned them, used GATK, which is just a tool to call variants. So I called my SNPs and my insertions and deletions, and then I annotated them using a software. So we can see here that there are tons of SNP differences and also insertions and deletions between bison and cattle, can we not? But even at this 22 and 24 million, how much percentage of that is 2.8 gigabytes? Like 1%? Fairly small. So we're still trying to focus on a small range. So the next thing that we have to do, or we're trying to do, is validate these SNPs because we have to increase our bison number, our sample size. 15 is good, but it's not good enough to make an integration test because you're going to miss SNPs. You have to increase the numbers. So that's what my old advisor is trying to do now. So I got to use my arrow diagrams again for Caprock Canyon State Park. It looks kind of like spaghetti up there, but there's a method to the madness, I promise. So Caprock Canyon State Park, again, was a closed herd for a while. It was supposed to be descendants of Charles Goodnight's herd. They started suffering from inbreeding and low heterozygosity. They were down to about 30% heterozygosity value. So Natalie Halbert, who I learned from, she did a paper showing how they are suffering from this inbreeding vortex and that it would basically be extinct if you didn't do anything in about 50 years. The herd would be dead. They're bringing in about 1 calf per year in a herd of about 50. So they were having very low calving rates, stillborns, miscarriages. And so they suggested bringing in outside bulls. So Ted Turner donated 3 bulls from his Castle Rock herd in New Mexico. And my job was to determine how those bulls increased heterozygosity in calving. So the first year that VC 1995 was used, he put in 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. He put in 11 calves by himself. So we're bringing in some outside genetics. We also considered, is this going to hurt bringing it? Are they actually going to do any good? So we looked at their heterozygosity rates as well as the alleles that they possessed at the microsatellites we usually test for. So the red up here are the founding animals that they had for Caprock Canyon State Park. The green basically is our Castle Rock bulls, and like each different color line is just tracing an animal to its offspring. And then the light blue and the different color blues is basically saying if it's an F1, F2, and F3. So the park can use this to see who still has original genetics, because we still have plenty of those in there that haven't resulted from an outside breeding. So that's what we kind of looked at for that. And now we're going to leave genetics behind. And graduated, if I wanted to be a professor or an instructor, I needed some more teaching experience because I wasn't writing the tests or grade. I was doing the grading. I just wasn't creating the tests to grade. So a little fun bit of information is most institutions or universities, community colleges, junior colleges, 4 universities, if you have a master's, you can teach. So that's a good starting point to always have a master's because I had that and I just graduated with my PhD and I taught microbiology lab. Dr. Jonas was my microbiology teacher and I loved it, but I hadn't looked at micros since that time, so I was a little worried. So basically I learned with the students kind of. So one of my favorite labs to do was this water lab where we tested water samples that was collected on the golf course at A&M. So we get to this eosine methylene blue agar plate, and I tried to tell the kids, if you see this shiny green, it's E. coli. I was like, you don't want that. This is why we don't drink stagnant water. And so some kids didn't believe me that I would actually turn this bright, shiny green. And about one of my lab groups each semester would have this. And so I would keep it for my other labs in case no one did, so I would show them. And they're like, oh, that's such a pretty green. I was like, yes, but pretty does not mean good in this situation. So then the hectone enteric agar plates is another fun one to do with them, and we actually did that with the water samples. So these EMB plates and the HEK plates are gonna not allow gram positive, only gram negative is gonna grow. So you don't really want a whole lot of growth on these. So E. coli is yellow here on this one, and then the main purpose for this is just to distinguish between Salmonella and Shigella. And so we can see here are different, I think my arrows got off a little bit because Salmonella is supposed to be this little black dot, kind of like it is here. And then Shigella is going to be this green one. But we would call those our Hawaiian plates because they have a nice little pretty sunset colors going on. But micro was fun to do, but it was only a 9-month appointment. So I was kind of looking for something to do in the summer and still maybe teach in 2016. So I went on an informal interview, it turned into a formal interview, and it turned into a full-time job that wasn't going to start until September. So I started my postdoc job with Dr. Saunders, and he is actually a clinician in the small animal clinic at A&M. He's an orthopedic surgeon. And so his main focus is cell biology. I'm in the small animal clinical sciences department now. We do canine mesenchymal stem cells, looking at characteristics and analysis of use in treatments. So this is a colony-forming unit plate of CMSCs. And so this is actually zoomed in because having to sit there and count all of those would be a little hard. So this is actually one colony that I zoomed in on pretty close. And so MSCs kind of give this spreading out, almost looks like neurons to me. And mesenchymal stem cells, the way we usually extract them is if a dog wants to have treatment using them, we'll extract from bone marrow the CMSCs. So we'll stick a little needle in there and aspirate the bone marrow, and then I'll grow stem cells from that. So they're not embryonic stem cells. You can get stem cells from your bone marrow, fat, tissue, cartilage. But these CMSCs, or the MSCs, are only about like 0.5% of the actual bone aspirate that we have. The good quality about these is these like to adhere to plastic. So whenever we plate them, they start growing, and then we'll wash using a saline solution. We'll wash off everything else and aspirate it, and those will stay behind until we use trypsin to lift them, and then we'll count them to see what we want to do with them after that. Osteoarthritis is a big thing for him because most of the cases he sees, being an orthopedic surgeon, have to do with osteoarthritis, hip dysplasia, and other fun things in dogs. So we also do construction of calvarial defects, which I'll go into a little bit more detail. Shape polymers with this, if you like physics, chemistry, biology, that might be your route. Biomedical engineers, because they created this little shape polymer right here. This is about an 8mm disc that we'll put into a rabbit. You heat it up in the water bath and it becomes almost spongeable, like you can move it to where you can place it in the defect, and then whenever it cools down, it'll harden. So we're trying to see if these shape polymers will help with healing time and also like make wounds stronger. Cartilage pellets, and I've just started doing histology. So I actually did it right. So this is actually immunohistochemistry on a collagen pellet, looking at collagen 2 and collagen 10. And we want this kind of purpley red staining, and these different pellets have different growth factors in them. So we're looking to see if the collagen levels are different just based on like visual representation. It's part of a master's project for one of his surgical residents, and she passed her defense in March, but they want to add this into the paper, so I'm trying to finish that up. But just different growth factors like fibro-FGF, which is beta-FGF, BMP2, and just looking at different dosages and see if that'll help with cartilage formation. And then qPCR and gene expression, 3D imaging, and we'll get to that, reconstructing the defects that we make, and then actually measuring like the bone volume and how much is actually grown back. And also doing that using densitometry, which is just looking at optical density due to exposure to light. So just some more about stem cells. So these are our little MSCs that we want, and these other little dots are things that we don't want. So whenever we wash, everything becomes kind of loose in this case. And this is actually where we lifted with the trypsin. So all of these are the MSCs that I want to capture for either freezing them for later use, or if we're going to inject, or if I'm going to do different differential assays with it. So after washing, you can see that pretty much there's still these little MSCs right there. So that's what we want. We want it to kind of look clean. And this is only at day 1. Usually we collect these whenever they get about 80% confluent. So from this one little guy, we're going to get a lot. So we typically don't want these to be fully covering the plate. because they're going to get clumpy. And if we're going to use these, we have to filter them for whenever so trying to squeeze these big clumps through a 0.2 micrometer filter is just going to get clogged. So we want the stem cells, but we don't want them to be in like just clumps. We want them to be nice and spread out. We don't want them on top of each other. So this we actually collected Monday or Tuesday. And so we froze these down because the new PhD student is going to do some ALP and ALZ assays, which is just differential assays. Okay, surgery picture. It's fine, I promise. So this would be a rabbit skull, and this is our 8mm calvarial defect that we created using like a little dental drill, like a tree fine. So for this experiment, we're going to make 2 holes, one here and one right across from the suture midline. And we're going to place the SMP into one of the defects. So this is placing the SMP, and this side will stay open. So some of our preliminary data is showing that our control site is kind of healing better, but as you can see, this is already starting to clot, right? If it's clotting, it's getting the nutrients. So this SMP, though, is kind of blocking from the stuff that to blood clot and get the nutrients in it. So the next step is to see, yes, there's healing in both, but how strong is one compared to the other? So we'll do some mechanical testings with it. And then this, you can see it's hardened, and it's pretty much perfectly in place. So these were our rabbits, and then we did micro CT on them. And so this is our SMP site, and then this is our negative control. You can kind of see these 2 little bone pieces trying to like go back in. So that's promising for our SMP, our shape polymer. So you can see the same 2 little points from this picture. This was given to us from the micro CT people, and then this is stuff that I used mimics on to create a bone volume measurement to see how much bone is actually in there, because you can't really measure it this way just by looking at it. So I created a cylinder and put it through these little holes, and it came back with these 2 images. And then you can create the full thing. It's just using different masks or bone measurements. So again, you can see how there's still, like, this is a good guide to have to see if your thresholding or your settings in the program are right, because you kind of want to recreate something that is similar to what the computer gives you. So then I overlaid one on top of each other, and so you can see that pretty much I got most of the bone that I was supposed to or could get using the cylinder. So we did the same thing in naked mice, but we only drilled a hole on one side and left the other side open. So that's why this is like complete bone, and then this is our defect. We put either stem cells, BMP2, which is a growth factor at high and low dose, and then stem cells with the growth factor in. And this I just got done on Tuesday. So this is our little mouse calvarial defect. And you can see here, and this is the recreated model that I made. I don't have a guide with this because we didn't send this out. We did in-house micro CT, so I learned the micro CT, which apparently is a very good skill to have. So if you like doing computers and imaging and micro CT and 3D imaging, you could actually get certified in it, and that's a very good skill to have. So this was using a faxitron, which is basically like an X-ray, to do densitometry on, and all of these are part of the same group. So this would be with our cells with BMP2, so our combination, and you can see after 4 weeks, this is only at 4 weeks, and so the rabbits were at 4 weeks too, that we do have some good healing in there. And then on our densitometry here, you can see the same thing, and we did groups of 5. So how do we find out our densitometry? So using all of our groups, so this is our mock group where we just put plasma and thromboplastin in. You can see that there's really not a whole lot of bone growth in there. And then this is our BMP high. This is our outlier. You can see like that's pretty much healed. And then the way that we measure is I put, using the software, 3 circles, and then we're looking to find a healing index, which is just taking different reciprocals and subtracting them from each other. So 1 over this volume that the software gives me versus the like this is our error to take into account error, and then this is our control side. So for the first 3, our healing index is only 0.138, so not very high. But so that's just how you can kind of figure out a healing index using densitometry. So getting towards the end, some other options that you guys may want to consider for life after college or wherever you go next. Of course, the professional programs, you know, med school, vet school, dental school, PT school, all those. One that I found that I had a strong interest in until I saw you have to get another master's was genetics counseling. So basically, we're learning a lot about genetic testing, genomic testing. Well, you get those results. Some people maybe don't know how to deal with that. So genetics counselors kind of help you deal with that. So you have to take some psychology and genetics courses, and it's a good program, it sounds like. I think U of H is the only one in Texas. I haven't looked lately, but at least whenever I looked a year ago, U of H had it. You also have to get board certified, so you have to take a certification test after. Pharmaceutical sales, my cousin's wife does that. She's very good at it. I am not the best salesperson in the world. My dad, great. Me, no. I know a lot of people who do laboratory supply sales for Thermo Fisher, VWR, Eppendorf. Of course, it's based on commission, but usually if you just have a biology degree and fairly knowledgeable in the lab and lab equipment, you can start off there. Laboratory manager certification, if you want to work, say, at like MD Anderson or Baylor College of Medicine, they usually ask that you have taken this test and gotten certified to be a lab manager. If you have a bachelor's of science. I know a lot of people who have a bachelor's degree and they're research assistants. They don't have a master's, but so you can do that if you really like research or if you want to get your feet wet a little bit more. Or specialized training, like in your bioinformatics, imaging, and histology is a big one. You can do histology, like in the vet school, people send their samples off, like for diseases to see if the dog has a disease or not, for histology. So there's just different options. My one suggestion would be, if you're in a lab, as long as you have good technique and you can follow the protocol and procedure, you can pretty much get trained in a different lab. Because again, I was genetics, genomics, PCR based, hadn't really done cell biology. And as long as you have good, steady hands, it'll be okay. So with that, I'll take questions. These are my dogs, but fun story. The black and white one, she went to all of my home games whenever I played softball, so she's 12. But yeah, she's the only one left from that time, but yeah. So I'll take any questions if you have them.
Questions for Dr. Dobson?
It doesn't have to be about my research. If you have outside question, yes.
So for the various bison herds around the country, is there any kind of plan for genetics of them, swapping animals or anything like that?
There's been some swapping already. Like most of the, so of the federal or the Department of Interior herds, you have Wing Cave, Teddy Roosevelt North and South. Well, Wing Cave started Teddy Roosevelt South, and then South made North with the help of the National Bison Ranger, another park. So things have been moved around already. And then the Henry Mountains herd in Utah, we did a paper on. and has very close relationship to Yellowstone because they're supposed to come from Yellowstone. And so that's another possible herd for maybe an aggression-free, although right now they're being grazed on the same land as cattle, but we didn't find any, to our best ability, we didn't find any integration, because it's hard to say that anything's integration-free because, like I said, 14 microsatellites compared to 24 million SNPs is a lot. So
most of the bison that are in private ranches are a mixture?
yes. Yeah. Yeah. Most of the bison that are around now are in production herds, so they usually don't have, because if you get down to it, if you do have cattle integration, it's 1% of your whole genome. So the main question I think to ask is what is changed within those, because we did a paper where we did two different kind of like feeding, like one was a feedlot and then the other herd was on Santa Catalina Island off of California. Not the best grazing conditions for bison. So we had feedlot, very good, and not good. And bison with bison mitochondrial DNA weighed more on average than bison with domestic cattle mitochondrial DNA. And so is it more important to just look at mitochondrial DNA and feed efficiency? Because if you're a producer, you want heavier animals. So that's something to consider. And then is genetic resistance, like to diseases in cattle integration bison, is that being affected by compared to an aggression-free bison? You just have to see what those genes are controlling, and it's still it's going to take a little bit to actually get all of that. Because there's a lot of bison out there to test. And the National Bison Association wants to get it up to 1 million. So it's yeah. Because the best idea would to make more satellite herds for conservation and everything, considering integration as best we can, heterozygosity, and that kind of thing would be the best idea. But also big enough land to carry the numbers. So.
I had a question about your master's degree, the King Ranch pedigrees you did. So when you were looking at like the inbreeding and then the genetic relationships, was that just sequencing of large portions?
Oh no, that was just looking at pedigrees, and then we use a software called MTF Rimmel. So basically, each animal in the pedigree that was traced back to Old Sorrel, I had to create a master pedigree file in Excel. I think I had about 1,300 animals in there, and so it's fine. So basically, computer programs. It was basically me using Excel and saying, yeah, it's mathematical and trying to figure out the genetic relationships that way. Okay. And so yeah. So we wanted to maybe go back, like whenever I started my PhD work, we thought maybe we could go back and do sequencing or microsatellites to verify pedigrees match that, but money really wasn't there. So, and then the USDA came up with the bison one, and he put me on that. So it worked out. Yeah.
Any other questions? If not, let's thank Dr. Dobson.
For more information, please visit tlu.edu.